Hello everyone, welcome back to our channel. I'm Dr. Ashmita. Let's continue with the GPCR, that is G protein coupled receptors series. In this video, we'll study about the G alpha Q signaling pathway. So as we have discussed in our earlier videos, whenever there is a GPCR receptor activation, this is a GPCR receptor getting activated because of presence of a ligand, it will transmit the signal to the G alpha subunit. Is transmitting the signal to G alpha, and the pathway will be chosen by the G alpha depending on this ligand. Either it can be stimulatory or inhibitory, or the Q pathway that we are going to study now depends on the ligand itself. So the ligands and receptors that are responsible for choosing this G alpha Q pathway are as follows. It is norepinephrine, a ligand acting on alpha adrenergic receptor, and acetylcholine acting on M1, M3 muscarinic receptors, histamine acting on H1 receptor, vasopressin acting on V1 receptor, and there is angiotensin 2 receptors. These are the examples of few receptors and ligands that cause G alpha Q pathway stimulation. So let's continue. So because of this signal transmission to this G alpha subunit, once it gets activated, it will dissociate from the beta gamma dimer and it will choose this Q pathway. On choosing this G alpha Q, now it will act on the G alpha Q will act on effector. Here the effector is phospholipase-C beta. We can write it as PLC beta. PLC beta is present just under the plasma membrane, that is on inner surface of the plasma membrane. Once this effector gets activated, it will cause hydrolysis of membrane phospholipids. Membrane phospholipids are PIP₂. This phospholipase-C beta got activated, it will act on the membrane phospholipid, cause its hydrolysis. PIP₂ will get hydrolyzed into IP₃ and diacylglycerol DAG. Let's see the full forms of PIP₂. PIP₂ means phosphatidyl inositol 4,5-biphosphate. IP₃ means inositol 1,4,5-triphosphate. DAG means diacylglycerol. So thus this phospholipase-C beta will convert the PIP₂, a membrane phospholipid present in the cell membrane, into IP₃ and DAG. Here, this IP₃, it is water soluble, so it will diffuse into the cytoplasm easily and it has its own independent pathway. Whereas the diacylglycerol, it is lipid soluble, that means it remains in the membrane. So let's first study about this independent IP₃ pathway. The IP₃ thus generated, it will diffuse into the cytoplasm and it will enter the smooth endoplasmic reticulum. This is a smooth endoplasmic reticulum. It is harboring, these are the receptors. It is harboring the receptors for this IP₃ molecule. Via these IP₃ receptors, the IP₃ molecules will enter the smooth endoplasmic reticulum and it will open the calcium channels for the smooth endoplasmic reticulum. These calcium channels are opened and there is like efflux of calcium moving out of calcium from the endoplasmic reticulum into the cytoplasm. So calcium will move out into the cytosol. Calcium is released into the cytosol. The released calcium, it can take two pathways. First, some of this will migrate towards the membrane. Some calcium will migrate towards the membrane, towards this DAG you consider. The balance, the other remaining calcium will bind to calmodulin, CAM. It is abbreviated as CAM. And this calcium and calmodulin will activate certain other kinases. It will activate further other kinases like calcium calmodulin dependent kinase. Secondly, myosin light chain kinase MLCK. So two types of kinases are activated by this calmodulin calcium complex. So the final output, the final result, because it is acting on this kinases, the final output is muscle contraction, secretion, metabolism, enzyme activation. This is the final cellular response given because of this signal. Now let's take an example. Whenever this alpha adrenergic receptor stimulation in the vascular smooth muscle, it will result in increased calcium release that will activate this MLCK, that is myosin light chain kinase, that will cause vasoconstriction. This is an example of this IP3 pathway. Now, coming back to the DAG, we have left the DAG in the membrane itself. It is lipid soluble, it remains in the membrane. Now, this DAG, it will act on a protein kinase C, PKC, present in the membrane itself. PKC, protein kinase C. So, it will activate protein kinase C with the help of the calcium also. Calcium from where? From the endoplasmic reticulum here. From the endoplasmic reticulum here, it will release the calcium. Some of this calcium now we have discussed that it is migrating towards the membrane, towards DAG. So both DAG and the calcium will activate PKC by binding to PKC. It will bind. The PKC has domains for this calcium and DAG. This DAG will bind on the C1 domain. It will bind on the C1 domain, whereas the calcium will bind onto the C2 domain. So C1 will bind to DAG, C2 will bind to the calcium. By binding to these domains, the PKC gets activated. Now it is activated. Now the active PKC, now it will diffuse into the cytoplasm and it will activate substrates. It will kinase means it will add phosphate to the substrates at serine and threonine residues. So we have discussed in earlier videos that in the G alpha stimulatory that will phosphate, that will add phosphate groups, plot protein kinase A will add phosphate groups at serine and threonine residues. Similarly, here also in this GQ pathway, in this G alpha Q pathway, here it is protein kinase C. It is protein kinase C that is adding the phosphate as a serine and threonine residues. That residues are present in receptors, ion channels, cytoskeleton proteins, transcription factors. These are the few examples of substrate upon which the protein kinase C is acting. The final response, the final cellular response is smooth muscle contraction, exocrine gland secretion, platelet activation, liver metabolism, that is glycogen is broken down. Because of glycogen breakdown, it will result in glucose release, glucose release, and there is immune activation and epithelial cell growth. Immune activation by activating the T cells, epithelial cell growth by stimulating the proliferation or differentiation. So this is the final output given by this DAG pathway. So IP3 pathway will give this output, the DAG pathway is giving this output. And finally, once the signal, the final response is given, the signal now it has to terminate to prevent overactivation. It is terminated by inactivating the PKC. You inactivate the PKC here. So PKC will be inactivated by either you remove the DAG from this domain, removal of DAG from this domain. Either it is metabolized or it will go back to its PIP₂ form. It will either be removed, it will undergo metabolism, or it will go back to this PIP₂ form. Or the calcium reuptake into the endoplasmic reticulum. Here in this pathway, calcium will be taken back by this endoplasmic reticulum here. Either this is the first termination is here PKC, second is the calcium taken back by the endoplasmic reticulum. Third is phosphatases that will remove phosphate from the PKC. From the PKC you remove the phosphate, so it becomes inactive. Inactive PKC means it doesn't act on this residues, target proteins. Or PKC downregulation. Downregulation is because of exhaustion. PKC gets exhausted after acting for a longer time. For prolonged activation, it gets exhausted and it gets downregulated. By these four mechanisms, the signal is being terminated to prevent overactivation. So what happens if it gets overly activated? What if the signal doesn't get terminated and the signaling is happening continuously, non-stop? So it will, with a clinical significance, will read whenever there is a smooth muscle contraction for a longer time when the signaling is happening, it will result in hypertension in the vascular beds and the asthma in the airways. And Liver metabolism increases. That means more glycogen breakdown, more glucose release. It will result in diabetes mellitus. And epithelial cell growth will promote epithelial cell growth. It will promote proliferation differentiation. Means it is an enhancer of tumor. So tumor promotion. This is the clinical significance related to overactivation of this pathway. Also, when it comes to this norepinephrine, when it's overly activating this alpha-1 adrenergic receptor, it will result in vasoconstriction. Because of this muscarinic receptors, salivary gland secretion occurs, and because of this histamine H1 receptors, there is bronchoconstriction and inflammation. And the vasopressin acting on V1 receptors will cause vasoconstriction and hypertension. Angiotensin II receptors also cause hypertension. These are the examples of few clinical relevances based on this pathway, G-alpha-Q pathway. So in our next video, we'll study about the G12-13 pathway. So if you find this video useful, please like, share and subscribe to our channel. Thank you.